sure. So again, I promised a very entertaining talk. Thanks for coming after lunch. So I like to call myself a quantum engineer. So this means that I build instruments to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us. So people get a little bit surprised when I say that I study things that happens inside birds, inside butterflies. So before I explain where I'm going, I really need to tell you where I come from. We're going to start talking a little bit, it's going to be short, about quantum mechanics. And by very fast, we are going to have talked about things of biological relevance, such as organismal migration, how our cells respond to oxidative stress, how our cells respond to radiation. And I hope I can convince you that I think we can actually learn with nature on how to build better technologies. And that's because I'm a quantum engineer interested in how quantum physics informs biology at the nanoscale. So I usually start my talk by saying that I think humankind is obsessed with measuring things better because this may mean that we understand them better. So you can think about measuring better frequencies. This is an atomic clock that sits at NIST, the National Institute of Standards and Technologies in the US, and that uses very precise atomic transitions to define very well what a second or what a hertz is. You can think about measuring better magnetic fields so that the image of a baby is better resolved. And yes, this is the magnetic resonance image of a baby inside the mother's belly. Or you can think about measuring better accelerations, for example, so that, say, your gaming experience is enhanced. All those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity that you want to measure is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can sense tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. And actually, you can prove that if you use quantum objects as sensors, quantum objects such as a single molecule, a single electron, if you use them as sensors, your measurement is improved. In other words, the sensor quantumness enhances your measurement. In my past, and I really need to talk to you about my past now, I've worked with a single electronic spin in diamond that could be put to use as a very precise magnetic field sensor. So here's the problem. A spinning diamond can be put to use as a magnetic field sensor. So what's spin? So spin is a fundamental property of objects, and it's as fundamental as mass and charge. In the same way that mass measures how much an object interacts with the gravitational field, charge measures how much, say, an electron interacts with an electric field, spin measures how much objects interact with magnetic fields. In the same way that charges can be positive or negative, right? And positive and negative charges interact differently with electric fields. Spins are usually denoted with an arrow, and spin up and spin down represent two different energies of interaction with a magnetic field. All quantum objects have spin, even if this spin is zero, meaning that they don't talk to magnetic fields. But one quantum object that have spins that people are very familiar with are electrons. So electrons have spins. So the particular electron spin that I played with in my past to put to use as a quantum enhanced sensor was a single trapped electron in the material diamond, like in the material diamond that some of you have in your rings, right? So this particular trapped spin, it has the very particular property that it lives trapped in the diamond lattice. If you shine light at this trapped electron, it absorbs the light, gets excited, and then emits light back. So if you are familiar with this, this means that this fluoresces, right? And what's very interesting is that the fluorescence of this the electron in your diamond, it's different if the spin is up or if the spin is down. And just by looking then at how strongly the diamond is emitting light, you can actually do what people in the mainstream quantum call quantum state readout. Just by looking at fluorescence intensity, if the quantum property of spin has one energy or another energy. So here's how it can work as a very sensitive magnetometer. Here you can see two different levels here in this vertical axis. You can take it as energy and say those two energy levels correspond, say, to spin up or spin down. And in the spinning diamond, their energy difference was about the microwave range, the same frequencies that you get from your microwave oven in your kitchen. And it turns out that one of these states, the lower line state, is insensitive to magnetic field. This means that if you immerse the diamond in a magnetic field, this level will not care. Whereas in the presence of a magnetic field, this other state is sensitive to it, meaning that this state, if we immerse the diamond into a magnetic field, will get promoted by a tiny quantity called delta. And it turns out that this delta, via something called Zeeman interaction, is proportional to the magnetic field strength. And what I'm going to tell now is actually extremely important, and it's a point that doesn't really get made, because this is how all quantum sensing works. Okay? The problem of measuring a magnetic field in this case is actually mapped onto the problem of the experimentalists know how to measure a mismatch delta from a known energy difference in the absence of magnetic field. Again, this is how all quantum sensing works. There are many other quantities, for example, electric fields, temperatures, that might also change this energy mismatch here, so that because the experimentalist knows how to measure mismatches from a known resonance, from a known energy difference, we can have information about that property of interest. And this is a very general principle. And again, now you've had a crash course on quantum sensing. This is how all quantum sensing works. But I won't talk about the but. I don't have time. So to cut a long story short, in my past, I worked with this tiny sensor that lived in diamonds that we could bring to very close proximity to a sample that produced a magnetic field, and we could measure then this magnetic field. So this is, I just presented to you a bona fide quantum sensor that actually works at room temperature and under messy conditions. The messy conditions of the diamond lattice, the messy conditions of the sample that it's in contact with. Now, turns out that I don't care about those sensors anymore. So, but up to say 5 years ago, this spinning diamond, those sensors that you see there, well, were the type of sensors that I was used to dealing with, all humankind made. But at some point, almost by chance, I realized that nature many times made sensors that outperformed humankind made sensors in crazy ways. So I'm here to talk to you about a magnetic sensor. I won't have time to make a lot of comparisons, but this magnetic sensor works in a very similar way to the technological quantum sensor that I just presented to you. And this is why I think that the future of biology is quantum. So let me describe to you this very particular nature-made magnetic field sensor that is quantum. But in order to do that, let's start talking about biology at the nanoscale, namely the chemistry. So it's been known for many decades that there is a class of chemical reactions that depend on spin, right? This property that I just described. This is how this works. There's a chemical reaction happening, and at some point, the chemical reaction comes to a crossroads. At that point, the chemical reaction effectively looks for the spin state of a particular electron. If the spin is up, the chemical reaction continues to one branch. If the spin is down, the chemical reaction continues to another branch. Importantly, the final products of those two branches are macroscopically different. So first of all, a tiny little finicky quantum property is actually, it's been known for test tube chemistry to affect biology big time. Now, if at the point when this chemical reaction comes to this crossroad, this spin sees a magnetic field, interacts with the magnetic field, in a phenomenon that is exactly the same as the technological quantum sensor, technological spin in diamond interacting with external magnetic fields, this spin in the chemical reaction is going to talk to the magnetic field, is going to do quantum sensing, and this magnetic field will alter the probability that the electron spin is up or the electron spin is down. It's in this sense that magnetic fields might also alter the macroscopic fates of chemical reactions. This interaction is actually very brief, but the consequences are again, felt macroscopically because the products are different and at much, much longer time scales because everything happens downstream. So this has been demonstrated in test tube chemistry at room temperature, in solution in the gas phase, in the solid phase, and for magnetic fields as small as the magnetic field of the Earth, which is 1 to 2 orders of magnitude smaller than the magnetic field that you sense when you put your cell phone close to your face. So here's how this goes from chemistry to biology. The reason why everything goes from chemistry to biology is birds, okay? So I really do not care about birds, and you're going to see that I really don't care about birds, but birds have the big advantage that they helped initiate the conversation. So birds have been known, again, for many, many decades, no mystery there, to migrate, when they migrate north and south, following at least as a partial cue, this tiny magnetic field of the Earth. And people tried many hypotheses on how might they be doing this. And none of those hypotheses actually matched with the experiment. Then in 78, a crazy theoretical biophysicist made the hysterically comical proposal that, well, maybe should the same type of spin-dependent chemical reaction be happening inside the bird under physiological conditions, birds and organisms in general could sense magnetic fields to the extent that they could sense the different physiological concentrations of products coming from the two different branches of the chemical chemical reaction. For example, this has not been neither proven or disproven, but one hypothesis is that, well, as birds look right or left, they see different, they experience different magnetic field lines, and maybe like in one line there's more of a certain product, like if you're interacting with this magnetic field, maybe there's more production of something that makes the retina of birds like less sensitive to light, and then if you look the other way, there's more chemical reactions producing things that make it more sensitive to light, so that maybe birds could see magnetic field lines because there might be some photomodulation of their retinal sensitivity. Again, this is not proven nor disproven. That's just a hypothesis, and that's part of the problem because birds are big creatures, right? But after this weird proposal in 78, experimental biophysicists started looking for animal, because birds, proteins that could sustain such spin-dependent chemical reactions. And at that point, the only animal, because birds, protein known to sustain spin-dependent chemical reaction was this protein called cryptochrome. So cryptochrome is a flavoprotein. It has a flavin a pigment that is, if you shine light at it, the flavin chromophore gets excited and it emits light, so it's a fluorescent protein. It exists in the retina of birds, in the antennas of migrating butterflies, but it's also present in all our human cells because it also has circadian rhythm regulation functions. And actually, after 40 years of research, magnetosensing based on cryptochrome, there's really a lot of evidence that cryptochrome might be involved in magnetosensing. Cryptochrome is, first of all, very widespread throughout the tree of life. All those organisms, from diatoms to birds to lobsters, express cryptochromes. And for the experiment, the organisms boxed in red are organisms for which there is some sort of correlative evidence that cryptochrome has to do with magnetosensing. Okay, so let me talk a little bit more about the available evidence. The available evidence for cryptochrome-based magnetosensing is actually very widespread, but unfortunately at very disconnected landscapes. I won't have time to go into details, but in this left part of the slide is some data that shows that for cryptochrome in solution, the electrons inside the cryptochrome are behaving as bona fide quantum sensors. Okay, so for cryptochrome in solution, there is no doubt that those things are behaving as quantum sensors when they interact with the magnetic field of the Earth. However, the next level of evidence occurs for like birds, for plates of cells, and the results of those experiments are mostly consistent with what one would expect from a spin model acting under the hood, exactly what we would get for test tube cryptochrome. However, it's very hard to say, well, the bird is doing its thing because there's a quantum stuff happening underneath. But let's go over this macroscopic evidence. For example, during migration season, people catch 20 migratory birds. Those are very complicated, beautiful experiments, but they're not enough, right? So they put them in cages, and then they see which way the birds want to migrate. And they mess up with the magnetic fields, and the birds want to migrate to another direction. Flies are not they do not migrate, but they can be trained to find food based on the presence of a magnetic field. The researchers do that, they train the flies, then the researchers remove the cryptochrome gene from the flies, they knock out the cryptochrome genes, the flies can no longer be trained to find food based on the presence of a magnetic field. In a further experiments, the researchers put back human cryptochrome inside the flies, and the flies are back to being able to find food based on the presence of a magnetic field. Again, most things here are consistent with the quantum, what's going on in the quantum level, but I mean, it's very hard to, I mean, it's those length scales are very disconnected. So here's our idea. What we are starting to do is actually connecting those two length scales. To cut a long story short, we are building glorified microscopes with coils where we can get one cell and try to look at quantum phenomena inside cells and try to study and importantly control those spins in biology as if they were, because they are bona fide quantum objects. We want to take a bottom-up approach towards actually, in a very quantitative way, refuting or confirming that what people see here is actually what's in here. And when I say I'm building glorified microscopes, it doesn't look like this. This is not our setup yet, but our setup will look like this. This is an atomic physics experiment. It's a big optical table that with electronics. So this, what we're building is really a quantum instrumentation applied to biological samples. Okay, this is actually something that is lacking in this emerging field of quantum biology. Experiments have been done predominantly by chemists and biologists, which is okay, but those experiments only provide correlative evidence in the sense that we need to go to the nanoscale to be able to, in the same experiment as we're planning, in the same experiment, try to control quantum mechanical degrees of freedom, read them out as if they were like in a quantum experiment, and correlate those quantum degrees of freedom to downstream physiological processes as seen, say, in regular fluorescence microscopy. So this is what we are after. I'll spend 1 minute talking about the fact that what's cool about this sort of things is that we have quantum mathematical models that can actually make predictions of what we can expect from our experimental results, which is a little bit different from usual biophysics efforts. We can use the tools of quantum mechanics called open quantum systems formalism to sort of model in either a complex or non-complex for us experimentalists way and predict how certain things are going to happen in the lab. For example, here's one prediction that we made. I'm dying to take this further experimentally, but here in this top right plot, you see the magnetosensitivity of cryptochromes in solution as a function of magnetic field strength for molecules stumbling in solution. The first thing that you see is that this magnetosensitivity is not monotonic with magnetic field strength. It goes up and down. And here, at about 10 times the magnetic field of the Earth, this magnetosensitivity is very much reduced. actually, this is well understood in general, and it's a general principle for reasons that people understand, that spin physicists understand. Those chemical reactions that depend on spin depend on tiny magnetic fields, on the magnetic fields on the order of the Earth, on the order of your cell phones. So this means that if you put your proteins, magnetosensitive proteins, inside an MRI machine with the 5 Tesla magnet, the 5 Tesla magnet is not going to change the balance of the chemical reaction. Those things only happen for small magnetic fields, which is good because you don't need a lot of magnetic field to affect biology, but it's also bad because that can be put to bad use if we learn how to control those things. The second thing that is very cool is that in this curve that is not monotonic, goes up and down, it peaks very close to the magnetic field of the Earth, which might be a coincidence, or it might be that there has been some sort of evolutionary pressure for the protein that helps birds to navigate following the magnetic field of the Earth to have evolved to work better, to work best around the field where it should. We are just instrument builders. We cannot tell apart if it's a coincidence or not, but we think that people who do directed evolution of proteins could actually try to see if we can apply evolutionary pressure to the design of such proteins, for example. But again, this is not a talk about birds. There is now a lot of more correlative evidence that the same type of spin-dependent chemical phenomena might be behind a lot of extremely important physiological processes. For example, the production of reactive oxygen species, that stuff that is good for signaling, and also like if you have too much, it's bad for you, has been shown to be regulated by magnetic fields in a way consistent with the spin model under the hood. Stem cell growth in planaria is also magnetic field dependent in a way that seems to be related to spins, right? Yield of DNA repair, cellular autofluorescence, possibly from flavin proteins in mitochondria. They're also magnetic field dependent in a way that is consistent with the spin model. But usually, the picture that I show to everyone that makes people understand what I'm talking about is this picture here. So I have a friend who's a precision measurement physicist. His name is Peter Füllinger. He's from Munich, and what he does for a living is building very good Faraday cages, very good hypomagnetic chambers that you shield external fields, and then he shoves ultra cold atoms experiments inside. That is like something very physical, doesn't matter. He started hearing things about, magnetic field effects in biology. So what he did, and now this experiment has been reproduced twice, one by a group at Tufts, the other by a group in Mainz. He grew tadpoles inside one of his hypomagnetic chambers under 2 conditions. So his hypomagnetic chambers has a noise level of about 1 nanotesla, which is already way weaker than 50 microtesla, which is the magnetic field of the Earth. So very, very tiny noise level. What he sees is that if during those 2 growth days, he applies a mimic magnetic field of the Earth inside with a tiny coil, the tadpoles are okay. But if he grows the tadpoles only under this nanotesla field, about, as you can see here, about 1/3 of the tadpoles are macroscopically malformed. Okay? Let me recap what I just told you. We're not even applying a magnetic field, we're taking out a tiny magnetic field that people don't usually care about, and you're creating havoc with embryogenesis. So because the field strengths involved are so small, if this were like a mechanism mediated by a big chunk of magnetite, you would need to have a big chunk of magnetite responding to the field strength. So everyone's best guess is that since such extremely large magnetite crystals have never been found in the tadpoles, everyone's best guess is that this has to do with spins. And again, what's very interesting, again, it's not only for cryptochrome. There are many proteins that are now known to be able to sustain spin-dependent chemical reactions. And actually, my personal guess is that it's something to do with melanin. Melanin, the stuff that gives it's melanin or melatonin? Which is melanin, the stuff that gives the color to our skin. It's also what gives the color to the tadpoles, and there's very rich physics in melanin. And again, this opens up, and I'm almost finishing, a Pandora box of questions, right? From space health, like space stuff. What's the magnetic field on Mars? Can we reproduce on Mars? Can we grow lettuce on Mars? To things such as, can we actually start to deterministically tune those endogenous quantum mechanical degrees of freedom, say, for theranostics? I am dead serious when I say that in 30 years, I want to take my phone. I want to say, well, today I have a wound, I need help with wound healing. You go to your phone, you click there, wound healing, and your phone produces the exact magnetic field strength and frequency that you need to do wound healing, right? So the technology is already accessible. What is not accessible is like how to get the quantum map, because each chemical reaction responds very precisely to magnetic field strength and frequency. And in order to get this quantum map, we need the type of experiments that we're gearing up to have. We need to have the experiments that treat those quantum mechanical degrees of freedom in biology as quantum objects, so that we can understand at the nanoscale, via the spin physics, how we can deterministically tweak things, right? So another thing, it's my last point, I promise. There is also evidence, like 2 papers, right? That's part of the problem, because all the data is correlative, but there's also data that shows that weak magnetic fields can alter the functioning of ion channels. Should it be true, that in a way consistent with the spin model, should this be true, that ion channel functioning can be tweaked via magnetic fields because of this quantum property of spin? Well, that's a whole new way of doing optogenetics. First of all, magnetic fields penetrate deep. Second, you don't need to do any genetic encoding. The proteins are already there, right? And the idea is that, I mean, it could be like, if you will, quantum optogenetics. It's a whole new way of affecting biology. It's not chemical, it's electromagnetical, right? So, yeah, so that's what we want to do. We want to use quantum technologies to harness and specifically deterministically control endogenous quantum degrees of freedom in biology. If you want to read my rant about how bad the funding situation is for this field, or why we should care about that, please read my manifesto called The Future of Biology is Quantum. This field is very misunderstood. There is no funding available for that, and we want to leave academia because our values do not match. It's a failed marriage. So if anyone is willing to fund us, because what I would like to do is have either a focused research organization or some sort of a startup where we can do in a controlled way those high-tech quantum experiments in biology, so that we can by design bring the field to the mainstream in the medium term. This is my wonderful team that got the idea of what we're trying to do, and I'll end by my usual way, by saying, may the quantum be with you.
Thank you so much. That was fascinating. So we don't have much time, but I do feel like we should at least take a couple of questions from you guys. So, okay, Shadi, go for it.
Yeah, I have one question about, you really caught me with the spin of, the spin state can affect chemical reactions.
Yes.
How would entanglement perhaps potentially be used?
This does not involve entanglement.
Okay.
So there are several levels of quantumness. Like everything is quantum because everything is made out of atoms. I mean, this is a little bit above that. This level is at the level where superposition is necessary. That's all that's needed. Entanglement actually is, if you will, it involves not only one quantum object, but more than one quantum object. This to work, like neither technological quantum sensors nor those natural quantum sensors represented, they do not need entanglement. And in fact, I'm not convinced that nature uses entanglement. I'm convinced that there's enough evidence to me that it uses superposition, but not entanglement necessarily. I think he
I
was.
think there was over,
Yeah,
over
quick
there.
question. I know that from the experiment with dead balls, I also watched or know about the work of Michael Levin from Tufts, and he's talking about bioelectricity.
Yeah.
So, can you explain how, whether it's like downstream, upstream, or how was the relationship?
So
there,
actually,
basically?
I'm in conversations with Mike because the spin people want to talk to him and his collaborators because we think that a lot of things that he sees and that he calls bioelectricity is consistent with the spin model under the hood. So that is something that I'm very interested in finding out exactly, but a lot of things that he calls bioelectricity, we think it could be...
It's just like a little different lens on the same basically mechanism. Maybe.
We think that the fundamental mechanism is quantum.
Mm-hmm.
It's not necessarily encoded in the voltages that he applies and stuff.
You had a
Yeah,
question,
it was-
Clarice,
Yeah, so this is totally new to me, maybe I missed something, but there are many cases in which humans are exposed to strong magnetic fields.
Yeah.
For example, by electric motors or stuff like
Yeah.
that. And yet, I'm not aware of a big epidemiological evidence that this is detrimental.
Okay, so first of all, what we care about and what messes up with biology is not strong magnetic fields, it's weak
That's the
magnetic.
curve that you were showing.
Yes, that is, I mean, it really, I mean, we really need to study what those things do. I'm not saying let's be alarmed, but someone needs to study, and no wonder why phone companies don't phone my work, right? Because for reasons that are well understood, the magnetic fields that mess up with biology are small magnetic fields, which makes it easy to affect with an app, but it doesn't take much to screw it up if you're from a foreign power with bad intentions. You don't need like a super magnetic field gun or anything. Yes?
To follow up on the question about the bioelectricity, many of those effects involve a long-term memory. And are you aware of any ways that for electron spin specifically, you can have a persistent state?
Well, persistency is something interesting, right? Because those interactions that I talk about, they're very brief, but their consequences are felt downstream and for longer time scales. The interactions themselves that mess up with the chemical reactions, they're like nanosecond to microsecond. But their effects, and that's the whole point of this, their effect might continue for much, much longer.
Just like a match basically, and if it burns, then it stays burned in a sense.
So just kind of following on from that, the instrumentation side,
Yeah.
that you were kind of building out your table.
Yeah.
Will it be start and end state, or are you hoping to capture the reaction trajectory and the biochemical pathways?
We can do, once those are built, we can do,
full end to end. That's phenomenal. Right, you can always reverse
Yeah.
engineer as well, and now you've got
Yeah, and-
biochemical pathway maps with the
Yeah.
triggered through.
that is the goal, right?
Yeah.
And we need to break the cycle of low-tech evidence for that, correlative evidence. We need to go to like causation.
Causal, yeah.
And the only way to do this is to build those tables. But there's a big disconnect because people who fund those experiments typically, they don't understand that to do my experiments, I need way more money and they take way more time to build, right, than typical biophysics experiments. But everything's absolutely on par with quantum experiments. Right? So this is my challenge and I think I should go.
Thank you so much. Yeah, we're going to have to end there. But thank you again.